aquarium at the Dauphin Island Sea Lab. My name is Mendel Graber. I am an educator here at the Dauphin Island Sea Lab, and this morning we are chatting with Natalye Bordelon. Natalye is going to chat with us about culturing cancer cells and maintaining them in the lab, and some of the things that you might use those cultured cancer cells with in research. So with that, I'm going to let Natalye tell you a little bit about that.
Thank you. So yeah, I'm talking a little bit today about cancer culturing at the Mitchell Cancer Institute is where my lab is. So I have a few pictures just to kind of go over the cells that I work with, a little bit of the instrumentation that I use. So first off, at the Cancer Institute, there's many different colleagues, and we work on so many different cells, but the particular cell line that I work on is bone osteosarcoma cells, which I have a picture of right here. So as you can see, this is a cancer cell with the cell membrane on the outside right here. We have it dyed where you can see the mitochondria and the nucleus of the cell right here. So that's just to show a little bit of what the cell looks like. Every cancer cell looks different that I work with. We all cancer cells use media and a certain type of apparatus to be cultured in. The type of cancer cells that U2S cells are, they're adherent cells. So we all work in a, just to keep us safe and to keep from contaminating the cancer cells, because they can get contaminated very easily. We all work in a Class 2 biosafety cabinet, which has all the instrumentation that we need to use in there. There's pipettes, and then we clean off all of our media and our cell vials and things before we culture them. So the cells that I work with, they're primary cells that we got stock from a company. I'm not quite sure. I came into the lab with them already having the stock of the cells. So what we do, we take a vial out of the freezer when I'm ready to use them for experimentation, and you have to keep them on ice and in liquid nitrogen, negative 80 degrees Celsius before you use them, because that's their optimal temperature for freezing. And then when you're getting ready to use them, you have to keep them on ice and quick thaw them in a heated water bath for 2 minutes before you put them into the cell culture media, because if you let them go too long, they don't like to grow if you let them thaw too slowly. So each cancer cell uses a different media, but they all have similar components. They all have growth factors that come from different animals, such as, like, we use fetal bovine serum for ours, which comes from cows. And then they have amino acids for growth, and then we have pH regulating buffering systems in there. They also have vitamins for their growth. And we also put in antibiotics, so just to further keep the cells from growing any bacteria or contamination along the way. And then typically how, for these cells, for U2S cells, they grow pretty quickly. I think the doubling time for U2S is about 24 hours. So if I'm just keeping up with the culturing of them, I generally do, like, I resuspend them in 1 ml of media to 10 mils of the solution, and I just watch until they're confluent or close to confluent. You don't want them to be overly confluent. And confluence means that they fill up the entirety of the petri dish that I'm using. So the optimal time to passage cells is around 80% confluent, because when they grow too much for the dish, they start to die, or they grow too slowly, or they start to develop different things. They can start to mutate if they continue growing in that same apparatus without being able to expand. So so how I passage these. So our cells, when they get, like I said, around 80% confluent, I rinse them with a a buffered solution, which we use a phosphate buffered solution because it maintains the pH and the osmolarity of like the body system. If we used water, then the cells would they would shrivel and they wouldn't produce as well. So we rinse them first to get rid of all of the growth factors and stuff that was on the media, because the growth factors actually affect the enzyme that we use to separate them from the plate, because these cells adhere to the dish. And we use trypsin, which is an enzyme that eats up the protein on the cell, on the outside of the cell, so it won't adhere to the dish, so we can move it to a new dish. So we have to first rinse it with PBS so that enzyme can effectively work. And then we patch a small number of the cells into a new dish and then let them grow for a few days. So I have a picture here. If I'm just doing regular cell maintenance, just keeping up the cell colony for however many days until I'm planning to do an experiment, I just eyeball it, how many cells I passage. But if I need to be more specific, like for an experiment, a certain number of cells need to be seeded, such as if I use these small plates right here, they only fit a certain number of cells. I have to use this instrument over here that we call the cell counter.
Can we go back to this? Is this a microscope slide with cells?
No, it's actually, it's a plastic plate, and it works in the same way that just a Petri dish works, except I can have 96 samples. It's very small. And we actually use the microscope slides with this instrument right here to do the counting, instead of having to do it the old-fashioned way and like put a slide underneath a microscope. We put a little bit of our cell suspension that we've gotten with the trypsin with a dye that we call trypan blue, and it dyes all the dead cells blue. So when we put this in the instrument, it counts it for us, so we can.
That's very handy.
Oh, super handy, because they're very small, and like in 1 milliliter of solution, there's up to 1 million cells generally. So it would be, not that we would ever count 1 million cells, but when we did it old-fashionedly, it would take probably an hour of counting individual cells within a certain square grid, and this does it for us, so it's very convenient. So that's pretty much how, oh, and then once we've passaged cells and they are in like whatever dish they're going to be in for however long for experiments or for just being in passage, we keep them in an incubator that we set to 37 degrees Celsius with a little bit of CO2 pushing through, just to mimic body temperature and body standards. So that's pretty much the gist of how we culture. And then we can go into a little bit of the experimentation of it now. So what I specifically use in a common experiment that I do with my cells. So the U2S cells, they're the base wall type cell, and for my experiment we underwent a transduction of the cells, which means that we incorporated a DNA into our cell that we manipulated with this program, and we grew it up so I could knock out certain DNA repair proteins for my cells. And this is kind of just explaining how those cells, once we how we got them. So how transduction works, and I don't really have a picture of it, but you have your cells that you've seeded using the Countess. We count out 100,000 cells into a dish that has 6 wells, and then we have our viral DNA that, in our case, well, in my case, had the repair protein OGG1 knocked out of the gene, and we incorporated it into that cell and let it sit for a few days. And it also had in that gene a resistance gene for, in our case we had hygromycin and puromycin, which are antibiotics. So what we do in that case, After we've grown the cells up for a few days, I passage them into another dish very sparsely, and I use puromycin or hygromycin, whatever was in the gene resistance, and whatever cells actually incorporated the DNA that we needed would stay alive, and the other cells would die, and we would be left with a very small colony of cells that we actually needed. And then we would separate them out with these little cylinders, very small, and use trypsin, and then we would have the DNA or the cells that we need to do our experiment. So for my experiment, we knocked out, like I said, OGG1 and this other gene called NUDT1. And for our experiment, how to make sure that we actually knocked out the gene that we were expecting to, we used a process called immunoblotting, which uses this instrumentation right here. So how it works, we take our cells that we've made that are missing our gene of interest that we have made up, and we go through this whole process of making a cell lysate, which kind of just separates it out into the protein that we need. And then
To lysate is to like rupture first.
Yes.
And so it releases the protein that you're looking for?
So it releases the protein that we're looking for, and we can store that in the fridge pretty indefinitely for several months. And there's this gel apparatus that goes into this chamber, and we pipette different, so I actually have an example of how it comes out when it's finished. So this has a marker of different molecular weights, and in each well, this is the wall type UTOS, and these are the two cell lines that are supposedly knocking out my gene of interest. So it shows that we successfully, when you undergo this, when you pipette them into this chamber, gel electrophoresis happens and the proteins transfer down the gel based on molecular weight. So these markers are at specific molecular weights, and my OGG1 protein is at 37. So we know that if the wall type, which will show OGG1, shows up and then it doesn't show up in the other two wells, that means we successfully knocked out the gene. So that's what that shows. And this is just another instrument that goes along with it. After you successfully transfer it down the gel, you transfer it onto a membrane, which you see here with that instrument.
So does this have a practical application?
Yes, it so not only is it used, it's pretty much used in that sense, like you are developing new cell lines, and it's a qualitative measure to make sure that you've actually done what you were supposed to do, like make sure that your gene of interest was knocked out, or in our case we also did overexpression genes. So it's just a good way to check to make sure that your transduction was successful.
So why do you have an interest in knocking out these genes?
So,
or generally speaking, why would there be an interest in knocking out these genes?
So my gene of interest is a DNA repair gene. So as you go throughout your day, your genome gets just damage that builds up on itself just through normal day. Every day the DNA is getting damaged.
So people might recognize this in a broad sense as aging.
Yes. And with aging it gets even worse. You have more DNA damage, but these OGG1 is a protein that initializes the repair of your DNA. So if there's a damage, it pinpoints it, and it's like, hey, there's damage here, we need to repair it, which is good in normal cells. In cancer cells, not so much. You want to kill off cancer cells. So that's why in chemotherapy, and that sometimes chemotherapy doesn't necessarily work because the whole point of it is to kill off cancer cells. But if we have the gene that's trying to go against it and say, hey, this chemotherapy agent is causing damage to the gene, we need to fix it, it's kind of going against the whole purpose of chemo. So if we have other therapies along with chemo, such as knocking out these repair proteins, it may help chemo be more effective.
And you can knock them out in the cancerous cells without knocking them out in the cells that are not cancerous?
See, that's where we have to do a little bit more study. I haven't gotten to the broader sense of it. You do it in cancer cells first, and then it eventually probably goes into test trials that I'm not a part of that test to see if it can be used as a therapeutic as well as just in the lab sense. So a lot of studies, it seems good on paper, like it'll work in just individual cells, but then when you move to an animal study, it may work still then, but then when you move further on to human studies, it doesn't work.
So to kind of simplify this, would you say that it's sort of like a high-tech, sort of like an assassin protocol for cancerous cells?
Yes, because if you knock out the gene that starts the whole repair process, there's other genes that may come and try to help it out, but my lab also kind of studies knocking out genes along the repair path. So if we knock out what starts it and it doesn't get picked up to be repaired with another protein, then it causes cell death, which is what you want with cancer cells.
Let's go back for a second.
Yes.
And go back to basics here.
Yeah.
And explain what a cancer cell is, because we don't want to give the impression that it is equivalent to a bacteria or a bacterium, like it's a foreign cell.
Yeah, so the difference between like a normal functioning cell and a cancer cell is that a cancer cell is immortal in that normal cells have a limit to how many times they can replicate, generally maybe 20 or so. It depends on the cell type and all that, but normal cells will stop replicating after a certain time. Cancer cells have mutations in their replication machinery, which...
So this would be a person's cell, not a foreign organism that has been introduced.
Yes. It happens to cells that are already there, and they mutate to get this machinery that they just continue to keep multiplying and don't die off by the natural just aging of cells like most cells do. And it happens in the body, but the damage that happens can occur from other things from outside. Like part of my experiment is testing different drugs that have been used in different therapies to just test and see if they work with this specific cell line. Also, I'm going to start touching into environmental hazards such as like pesticides and flame retardants. And
so there are environmental things that can trigger or stimulate these mutations in a person's cells.
Yes. So if we understand how the cell path, like the repair function works with just the base damage and with like normal damage caused from like chemotherapy and the like of that, then we can eventually branch off into seeing how environmental stimuli affects cancer growth. And
I think people are pretty familiar with, the observable effects of cancer. But can you talk about that at a cellular level? So what is going on with these immortal cells, a person's cells that have mutated to become immortal in their replication? How does that at a cellular level cause problems?
So with normal cells, like I said, it has the normal machinery that will repair damage, and it will replicate pretty effectively. There's a lot of components within a cell that allow replication to go smoothly. With a cancer cell, it's constantly, if you get one mutation that makes it past the repair proteins of sorts, it can continue to mutate and then just slightly run rampant in sorts. It can form tumors, and they form in the bone. So it's there's just a cell that it just overrides the normal factor of repair and normal replication of DNA. It's instead of having the normal DNA strand with what's supposed to be there, it has mismatches of different damage and like different bases that aren't going where they're supposed to be.
So it's not performing the functions that kind of cell should be performing, and on top of that, it is the population of this cell that has these malfunctions is increasing at a rate that that can then cause problems if the functioning that it's supposed to be doing stops because it's replicating.
That's what's going on.
So one of the things that you said that kind of I just want to clarify this. You mentioned staying safe in the lab, and I don't think of a cancerous cell as like contagious per se, but if a cancerous cell, so if you somehow got a cancerous cell into your body, would that have the effect of, as you were saying, there are some environmental things that can stimulate your cells? Would it function that way?
I don't believe so. I actually haven't thought about that. When I was saying that we protect some of the things that we work with, like some of the drugs that we work with are harmful. They're carcinogenic themselves because they're for killing cancer cells. And then also when we're doing our transduction experiments, we work with virus, which is very I lost the word. Very infectious.
Yeah.
So I don't think that like getting cancer cells on you would matter, but also the cancer cells stick to the plate if they're alive. If they're dead, then they float in the media. So you shouldn't get them on you unless they're all floating in the well, which.
And are the cells you're working with human?
Yes, these are human cells.
And so I think that there are some people who, and I mean, I don't know this, but have had cancer removed, like through surgery. So, you might have chemo or radiation, different kinds of treatments for cancer, but one treatment would be surgically removing cancerous tissue. So if that cancerous tissue if the cancer is like disrupted in the body physically and it's not all removed, does that increase the risk that it spreads throughout the body?
If it's not all removed? Yes, it may hinder, because the more cells that there are, the faster they're going to populate. But if you leave just one, there's a chance that there's something in the body that can attack it and kill it, but there's also a chance that it can continue to replicate. There's, with my cells specifically, the ones that we've mutated to like have this gene response, we constantly have it in a media that they're supposed to be resistant to, but even after passaging it over and over and over again, they themselves can mutate and have different things than like different things than what we were trying to see in the beginning. So when you're doing cancer experiments also, you don't want to use the same cell line well past its passage. You use it up to like 15 or so passages, but after that it can start mutating in itself even.
So can you explain again what passaging means?
So how we like maintain cells, we have them in a dish, and a passage is pretty much just taking a small portion of whatever's in this dish and moving it into another dish so it can continue to grow. And that would be pretty much the second generation of that cell.
And when you say grow, you mean the number of, it's replicating, so the number of cells is growing. And so this is kind of, this is a strange perspective that you were trying to keep cancer cells healthy in order to experiment with them. So would you say that that's kind of a fair way to describe it, because you are trying to culture and maintain these cancer cells and understand their survival, and then ultimately, tamper with them, so that you can figure out how you might be able to target, assassinate them, sort of, to target them for, so that they can't replicate.
Yeah, that's exactly what we do with them. We keep them alive until we do cell death experiments to see what happens and what dose of different drugs or environmental toxins affect them, or if it kills them at all. Because sometimes it may not necessarily kill the cell because the gene, or there's another gene that's repairing the damage and letting it live, but other things are happening too. So it's still living, but mutations are still happening. So a lot of different branches that happen with these cells.
So you are isolating DNA that is meant to, your intention is to introduce this into the cancerous cells because as they replicate, because, right?
Mm-hmm.
Because it then inhibits their ability to
repair. Mm-hmm.
And you're doing this using a virus.
Mm-hmm.
Okay, so can you kind of explain that again?
Yeah, and I'm not, this is one that always kind of I trip up on too, so I may not have the best explanation for it, but so there's a program, and I lost the name of the program. There's a website that you can order and design different viral vectors of DNA, mm-hmm. and that we incorporate them into a stable cell line that then introduces virus out into the media, which we can put on our cells of interest, and it can, it's called transduction, and it can infect them with our DNA that we want. And this DNA also has, as I mentioned before, it has the resistance gene for whatever antibiotic that we use. So if those cells successfully absorbed the DNA from the virus, then when we dose it with media that has that antibiotic, they should survive, and any cells that didn't successfully get the DNA die off. So that's why we have our cells that then show this example, like we did the same process for these OGG1 knockouts. So the wall type has OGG1, and then the other two cells don't have it anymore.
So where did that DNA come from? So this DNA has both, it has both code for the, it doesn't have the repair, and also it is antibiotic resistant. Mm-hmm. So where did that DNA come from?
We ordered it online.
So some researcher in the past has isolated this, and it's reproducible so that you can experiment with it.
Yes. Mm-hmm.
And so then you want to separate out the effects. So do you understand why some of the cancer cells would would be affected by that viral transfer of the DNA, and some of them wouldn't?
It may just be that the media didn't reach them as well. It may have been highly more concentrated in one area, because we have the cells all grown on a plate, and then we drop it like this into the media. So it may just be after, because we only let it sit on the cells for 1 or 2 days. So it could just be after 1 or 2 days, it really was highly saturated in this one section. So those cells were able to have it in the media and actually absorb it. There's other components in the mixture that make it where the cell, the cell wall, like the cell membrane, it allows transfer into it. So it could just be that maybe some cells are more resistant to it than others. For the most part, with UTOS specifically, they're highly transducible. So they pretty much take up whatever foreign DNA that we put on them. Other cell lines are different, and I don't know a whole lot about them, but I know that's why I work specifically with these, because we knew that we were going to be modifying them, and they work a lot better than other cells.
So this is a step in a research process
Yes.
that may eventually have some practical applications for cancer treatment. So just to kind of clarify, in a big picture, what might be, what might have some practical outcome out of this.
So I'm hoping with my experiment that I'm eventually going to be testing different pesticides to see how they affect my cells, and we will see that if there's another experiment called a comet chip that shows DNA damage. You could actually quantify DNA damage. So we're hoping to see that you would think that DNA that doesn't have a repair gene would show extreme damage, while I have another cell line that has excess of the repair gene. It should show minimal DNA damage in comparison to the wall type one. So we're hoping that if we dose it with different levels of environmental toxins such as pesticides, we haven't completely decided on what environmental toxin that we're using. If it sticks with this trend that my knockout genes show a lot of damage and then the overexpressed don't show a lot of damage, that the damage type from that, the damage that that kind of environmental agent causes is oxidative in nature. If that, it's kind of hard to explain. Like my repair process is an oxidative repair process with this gene. So if it doesn't follow that trend of repair, then this is causing human health damage, because we all know from several studies that pesticides and herbicides cause damage in the environment to different, like marine life and that sort of thing.
But why would you be using cancer cells as your experiment for your experiment with the pesticides instead of healthy cells to see if that causes them to become cancerous?
Cancer cells are easier to manipulate.
Okay, so that's the reason, because you can produce the ones that have the greater or the lesser repair DNA.
Yeah, healthy cells die off too quickly. They only have a certain amount of time that they replicate, so you can't continuously use them. For my cells, I can freeze back several vials worth and just continuously use the ones that I've manipulated.
It's interesting that you can freeze these living cells and then...
It is, and it's weird how specifically frozen they have to be, if that makes sense. Like I once had them in a fridge that was minus 80 degrees, and they didn't live as well when I unfroze them as liquid nitrogen frozen. So I was like, just slight temperature variances affect
And these
them.
protocols are already established, the ones that you are using to grow and maintain your cells?
Yes, they've been over and over again optimized so to be least infectious to the experimenter, because viral transduction has been used for ages, but it used to be very dangerous. You had to use like hazmat suits and different levels of safety cabinets, but now when you're doing viral transduction, they separate out the infectious part of the virus until like the very last minute, so you're less handling of it.
So that's one of the things that I like to kind of repeat so that people hear this over and over again, is that your experimentation kind of gives you it will give you a piece of the puzzle, and then that piece you can add to things that other researchers are pulling together. And so we advance this way, because some people like to know the practical applications of research, and so you are building on some protocols and some technologies and some products that researchers have already put together, their own small bits of the puzzle, and then you add more to that with your experimentation.
It's
right?
very interesting to see just how it's progressed from the beginning and then to how it is now. Like the fact that I don't have to sit at a instrument or at a microscope and count individual cells, and I can just plop it in there. And it's time
Yeah, that's
saving
another
and-
thing. So, do you know how much this instrument costs?
I don't.
Because there are still researchers who are counting cells, and I mean, we just during the last Boardwalk talk, we talked to a researcher here who's counting algal cells, and so if this instrumentation exists for different kinds of cells, and if it's accessible cost-wise to researchers, it kind of changes the game as far as the amount of time that you are spending on that versus doing something else. That's interesting.
Oh, most definitely. I've definitely been blessed that I didn't have to hand and sit and count that, because my efforts are elsewhere, like individually pipetting 96 wells. So give and take, if I have to spend less time counting my cells to then use for an experiment, then I'll have more time to dose individual plates, which takes some time.
And so if we go back to what kind of practical applications these might have, we might offer that as a like a justification for the research. So if you continue this and you are experimenting with environmental toxins or pesticides or different kinds of things like that, then you might be able to show how healthy human cells might be vulnerable to different toxins, right? So that would be maybe one application.
There's different assays that you could look at, which are experiments. Like one that I've looked into at different concentrations of. I haven't went into environmental toxins yet, but I've used just different combinations of therapy drugs, and at varying concentrations, I could show the growth trend of how more of a drug affects it with these plates. Like with the 9612 plate, I started a low dose, and then as I continue going on, you can see in that plate how it grows, if they die, if different concentrations of drug affect it. Or you can even take a plate like that, put it into like I mentioned before, a comet chip assay, and see how different doses is causing DNA damage, which ultimately leads to cell death and other
So
issues.
you can optimize the dosage. You can make it as more specific, so that you don't have to use that you're getting a high enough dose without making it too high.
Yeah, that's typical experiment is to start off doing a dose trend to find like you don't want to use a dose that automatically kills everything, and you don't want to use a dose that's not showing any effect. So middle of the way dosing, and you don't know that until you throw it on a plate, and it may take several times. You may automatically start with doses that are too high, and you have to cut them down to smaller doses. And
Is there a lot of variability between the dosing for different types of cancer cells? So would this kind of experimentation have to be done with all the different kinds of cancerous cells, or is it more general?
It's very specific. So that's another thing. You never know how one cell's going to be affected by one thing over another. I'm utilizing a experiment that used one set of cells, and they got this completely different result than me. And I was like, oh, I guess it just doesn't affect U2OS cells in the same way.
So can you remind us what kind of cancerous cells you're using?
This is bone osteosarcoma cells, so they're bone cells. Typical experiment, these are just very stable. They grow, they don't die off as easy as some other cells, because even though they can multiply indefinitely, you still have to make sure they have food, like the right kind of nutrients and stuff to grow. They can last a little bit longer, and they grow a lot better than some other cells.
And so that would be true for the, coming to understand the environmental exposures too, wouldn't it? So, I mean, everybody knows, for example, smoking can cause lung cancer. And they associate smoking with lung cancer rather than bone cancer or other kinds of cancers. So the environmental exposures, the kinds of cancer that they might stimulate would be different too. So that would be kind of putting together that piece of the puzzle as well.
That's why we're testing to see if the environmental toxins even affect these cells, because they may show no trend of affecting them.
For the bone cells. So you've got a couple of different potential practical applications that you've explained to us, and then maybe farther down the line, with some further research, this whole idea of targeting the cancer cells and disrupting their, or taking out their repair gene might allow targeting of these cells, but you're not sure whether you could kind of target that without targeting the healthy cells. Or, I mean, you were talking about how the virus will, it will affect some of the cells and not others, and it may just be like the way they are exposed. So then that would be maybe another area of research would be if that works to target the cancer cells and not the healthy cells, then how to administer it as a treatment.
There's a lot of questions.
Yeah.
It's and that's why we study it, to see if there's ways that we can possibly answer all these questions. But they're so far I haven't gotten there quite yet.
Well, we're just talking about maybe what kind of big picture, like I said, kind of justification for the research to put that in context for folks, and also eventual practical applications, so those are related and kind of putting it in a broader context of research. So how long have you been working on this?
I've been working on it on and off I'm in my third year of graduate school. COVID has kind of messed with the timeline a little bit, but about 2 years.
Okay, so you've got your established protocol, and you've been working on growing and maintaining your cancer cells and isolating out the ones that you have incorporated the DNA to knock out their repair gene. So then what kind of pesticides are you testing on them?
We haven't completely decided yet what we're looking into, because I mentioned pesticides and he was like, we could even use like different heavy metals. Like they have like arsenic and lead and those as well. There's flame retardants like TBBPA that I was looking into. So that it's broad. We are still in the discussion phase for that. Okay.
And then, so there are several different ways that people might be exposed to those different environmental toxins too. So some of them would be like skin contact, where they could absorb it through their skin. Some of it would maybe be inhaled. Some of it might be ingested. So, would any of those, like kind of like the pathways of introduction into a human body, affect the, how you would select the toxins you're going to test?
Preferably we were looking into ones that were already, not necessarily ones that have already kind of been phased out, but the ones that other research has said are on the chopping block of we needed more research into whether these are carcinogenic or how they can affect different cells. So it doesn't necessarily the uptake of it that we're looking into, just more of a environmental sense, like which ones are showing to be an issue so far. And then we also need to look into, and haven't really gotten that far yet, of course, someone that's not in close contact to these pesticides or flame retardants or whatever environmental toxin that we decide on, would have less of a reaction than someone who works with it constantly. So it would probably first go through workplace hazard like tests first before we branch it out into, we work with those high concentrations that those people would be introduced to then just every day.
So in order to select the toxins that you are going to test, to research, you might first eliminate the ones that have already been researched a lot because that's pretty well understood, and then you might select some that that people have, that there are some people who have exposure to, that you know they're exposed to it. So that would be like a couple of ways that you would look at selecting which ones to test.
And we are trying to find ones that there is some evidence in that it causes DNA damage specifically.
Okay.
Like if it causes some kind of DNA damage, then we want to look into it, because generally most of the studies that I've come across thus far have not used my specific cell line. So it's good to have just broad, they tried it on this cell line using this approach, I could try it with my approach with my cell line.
So you have kind of a big message to leave folks with about this kind of research, or specifically about your research?
I think my research is really interesting, and just want everyone to understand that cancer biology and cell work in itself is very broad. So it's not something that one specific study, or it could just be cured overnight. There's a lot of different components that go into it. So there's a lot of different people working on it and just trying to find ways to help and work on it.
Well, we appreciate the research that's going into it and increases human health. And thanks for chatting with us about it this morning.
Thank you for having me.
Thanks for joining us.